The steps of preparing roots for mitosis divisions are: 1. Make roots pretreatment, in ice water for 24 hours. 2. Cut a small piece of root off and use forceps to place it in the cup of 1 normality HCl for 4 minutes at 60 Celsius. 3. Transfer the root into the cup of acetic alcohol fixative solution. Then, cut off the bottom 1 or 2 mm of the root tip and discard the rest. 4. Gently lower a cover slip over the root tip. For squashing, without twist the cover slip, by pressing on the cover slip with your thumb, to spread the cells into a single layer. 5. Allow the slide to dry. 6. Place your prepared slide on the microscope. And 7. Use the 10x power objective, on your microscope, to look for thin layers of cells, and then, use the 40x power objective, to observe meiotic stages in individual cells. As mentioned in the next 2 slides.